It's for me a pleasure to introduce all of you today to Elena Sánchez. Elena got her PhD in 2017 from the Faculty of Pharmacy and Food Sciences at the University of Barcelona. She's currently a lecturer professor and has been recently awarded with the Professor Requalification Grants and is undertaking a stage in the Institute of Advanced Chemistry of Catalonia. She has carried out several stages around Europe, published over 80 papers, and supervised 2 doctoral theses. Elena is part of the CIBERNED group of Professor Antoni Camins and belongs to the research group Nanostructurated System for Prolonged Drug Release of the University of Barcelona. Her main research interest lies in the development of nanocarriers encapsulating active compounds for a wide variety of pathologies, including neurodegenerative diseases. And her talk today is devoted to biodegradable nanocarriers for the treatment of neurodegenerative diseases. So Elena, welcome. Thank you very much for your willingness to share with us your work. And the floor is yours. Thank you.
Thank you, Dr. Medina. I'm going to share the screen. Okay, and let me see one moment. Okay, can you hear me properly and see the presentation, right? Yep. Okay, perfect. So thank you, Dr. Medina, for your kind introduction. And I'm going to start this presentation about biodegradable nanocarriers for the treatment of neurodegenerative diseases. This talk will be divided in a first section that will be the introduction, then the objectives of the project, materials and experimental, and finally the conclusions. So basically, neurodegenerative diseases consist on the progressive loss of function of the neuronal cells, and it's calculated that they affect more than 1.6 of the population of the European population. It's also calculated that by 240, they will be the second cause of death. Among several neurodegenerative diseases, Alzheimer's disease is the most common cause of dementia. It's so common that it's calculated that every 70 seconds, a case of Alzheimer's disease is diagnosed in Europe. Alzheimer's comes with a lot of symptoms, such as confusion with time and location, difficulty in solving problems, and the well-known memory loss. All these symptoms come from a cognitive dysfunction that it's originated at the brain level. And the main hallmarks of Alzheimer's disease are the extracellular deposition of amyloid plaques, as well as the intracellular neurofibrillary tangles. Also, it has been observed that there is a chronic inflammation associated with neurodegeneration and with Alzheimer's disease. However, there is no current treatment that is effective curing this disease, but we have 4 approved drugs for this pathology. We have approved 3 inhibitors of the acetylcholinesterase, which are donepezil, rivastigmine, and galantamine. And it has been approved last group of drugs that it's memantine, that it's an antagonist of the NMDA receptor. In addition, there is another neurodegenerative disease that affects the ocular level that it's glaucoma. Glaucoma is the second cause of blindness worldwide and affects people over 60, 70 years old. Specifically affects 15% of the people among above 70 years old. What happens in glaucoma is that the patient, as you can see on the slide, the patients suffer a reduction of the visual field that, if it's not treated, it finishes in blindness. We don't have, in the same case as Alzheimer's disease, we don't have a cure yet for glaucoma, but we don't have a cure yet for glaucoma. What happens in glaucoma is that actually the drainage channel that eliminates the aqueous humor, it's blocked, either partially or completely. This makes that the aqueous humor gets accumulated in the eye, and this causes an increase in the intraocular pressure. This increase in the intraocular pressure damages both the retinal ganglion cells that are located on the retina as well as the optic nerve, causing the reduction of the visual field that we have seen before. In addition, the treatment of glaucoma doesn't cure the disease, and it consists on some eye drops that are able to decrease this intraocular pressure. And for severe cases, laser or either a trabeculectomy can be performed. Therefore, here in this work, we have studied two different drugs. The first one was memantine. Memantine, as we have said, is a drug that is already approved for Alzheimer's disease for moderate to severe cases and is an NDA not competitive antagonist. This drug is approved for Alzheimer's disease, but not for glaucoma. Specifically for glaucoma, there was a pharmaceutical company that carried out a clinical trial using memantine. And actually, they spent a lot of money and a lot of years as researching memantine for glaucoma. And finally, on phase 3, the clinical trial showed that the results were not significant. However, there was some improvement of the patients, but the results were non-significant. This gave us the idea that it's possible that memantine worked for glaucoma, but was not able to fully arrive to the retina. That is where it should be active. In addition, as we have said before, in Alzheimer's disease and also in glaucoma, it has been observed that there is an inflammatory component that causes the neurodegeneration, that contributes to neurodegeneration. Therefore, here we postulate that anti-inflammatory drugs such as NSAIDs could contribute towards an improvement of both Alzheimer's disease and glaucoma. Therefore, we have studied dexibuprofen for this kind of diseases. Dexibuprofen is the active enantiomer of the well-known ibuprofen, but it's more active than this drug and possesses less side effects. However, we have a problem because in order to treat both diseases, the drugs have to cross two of the most restrictive barriers of our body. The first one, in order to treat glaucoma, is the blood-retinal barrier that is on this posterior eye segment, and at the brain level, the drugs have to cross through the blood-brain barrier. Both barriers are highly restrictive, and actually, they expel the majority of the drugs. That's why we are studying nanostructured systems. There is a wide variety of nanostructured systems, but in this work, I'm going to talk about PLGA nanoparticles functionalized with polyethylene glycol chains. The advantages of these nanocarriers is that they are able to encapsulate the drug inside, and they are able to protect the drugs against metabolism and ionization. In addition, they are able to direct the drugs to the target tissue, crossing both the blood-retinal barrier and the blood-brain barrier. These systems, of course, they are biodegradable and biocompatible. At the ocular level, they are able to increase the ocular retention, and they are able to obtain a prolonged drug release, which actually increases the therapeutic adherence of the patients. So basically, the aim of this project was to encapsulate either memantine or dexibuprofen into PLGA nanoparticles covered with polyethylene glycol chains. All this work raised 4 different studies, 4 different papers that you have here on the slide if you have more curiosity. So basically, what we did in order to obtain these nanoparticles is, first of all, we had to prepare these nanoparticles. And for the case of memantine nanoparticles, we used the double emulsion method. Very briefly, the double emulsion method consists on solving memantine that in this case was water soluble, in a water phase and adding this water phase to an organic phase containing the polymer. Both of them, this mixture underwent to ultrasonic energy in order to form a first emulsion, and then a second water phase containing the surfactant was added, and this again underwent to ultrasonic emulsion. Finally, the organic solvent was eliminated, and we obtained memantine nanoparticles. A different procedure was used for the ibuprofen nanoparticles, and we obtained them using the solvent displacement method that briefly consists on solving both the polymer and the drug in an organic solvent and adding this mixture into a water phase. Afterwards, this acetone, the organic solvent, was evaporated under reduced pressure, obtaining these nanoparticles. Once we have prepared the nanoparticles, we also studied the optimization of their parameters. So basically what we did is we optimized each one of the parameters involved in the formulation in order to obtain the best formulation possible for the treatment of neurodegenerative diseases. Just as an example here, I show you the factorial design that we use in order to optimize the sonication parameters involved in the preparation of memantine nanoparticles. Basically, we studied how the wave amplitude and the first and second sonication time influence the properties of the nanoparticles. We studied how they affect to the size of the nanoparticles, to their polydispersity index, which is implicated in the polydispersity of the nanoparticles. We also studied the zeta potential, that the zeta potential is implicated in the stability of the nanoparticles. And we also studied the entrapment efficiency, because of course we wanted the maximum entrapment efficiency, which will mean that the maximum amount of drug is loaded inside the nanoparticles. When we studied these parameters, we obtained some trends, and as an example, one moment. Okay. I think I'm gonna pause it one moment. One moment, I'm gonna share again. Okay. 1 minute. Okay. Okay, here we go. So basically, as an example, we obtained some trends that the nanoparticles follow. And as an example, I show you a surface response that we have obtained, where you can see the influence of the amplitude and first sonication time on the polydispersity index of the nanoparticles. As you can see here, we want to obtain low polydispersity index, which will mean that our nanoparticles are homogeneous. And in order to do so, we need high amplitudes and lower first sonication times in order to obtain monodispersed samples. Well, studying each one of these parameters, each one of these parameters, we obtain nanoparticles with an average size below 200 nanometers, a polydispersity index below 0.1, a zeta potential highly negative, which will mean that they possess a good short-term stability, and an entrapment efficiency of more than 80%. Also, we wanted to observe the nanoparticles using microscopical techniques, and in order to do so, we use atomic force microscope and transmission electron microscope. And as you can see here on the slide, we saw that actually the nanoparticles had a round shape and a smooth surface, and they were not aggregated. We also wanted to confirm that actually the systems that we have developed were able to give a prolonged drug release. And in order to do so, we carried out a dialysis experiment. Very briefly, this dialysis experiment is carried out by placing the formulation into a dialysis bag that is composed of a membrane that is semi-permeable, which means that actually it lets the free drug cross through, but not the nanoparticles. So we did what we did is we put either the nanoparticles or the free drug inside this membrane, and we collect samples at different time points, and we measure the amount of drug that was released. We plotted these results, and as you can observe here on the slide, we saw that actually the free drug, in this case memantine, was released much faster than the drug encapsulated into the nanoparticles, that is the pink one, that are memantine nanoparticles. In the case of memantine nanoparticles, a burst release was observed corresponding to memantine that was on the surface of the nanoparticles, followed by a prolonged drug release of the memantine encapsulated inside. Moreover, for the glaucoma experiments, we also carried out ex vivo permeation studies, and we did, in order to do so, we used the France diffusion cells. That basically, it's a really similar, the mechanism is really similar to the one of the dialysis, but we place the formulation on the upper side on the donor chamber, and this donor chamber is separated from a receptor chamber by a membrane. This membrane was either the cornea or the sclera of a New Zealand rabbit. As we did before, we also extracted some samples from this receptor chamber at different time points, and we measured the amount of drug that permeated through either cornea or sclera of the rabbits. Actually, by doing these experiments, we confirmed that our nanoparticles show some corneal tropism, and the drug was able to permeate in a higher amount through the cornea than through the scleral tissue. Finally, we evaluated also the short-term stability of the nanoparticles at different temperatures, and in order to do so, we used the Turbiscan lab. What the Turbiscan does is we put the nanoparticles inside a glass vial, and the Turbiscan scans the sample, and we carried out this scan once a month, obtaining the backscattering profile of our samples. We measured this backscattering profile once a month during 6 months, and we observed that actually the nanoparticles were stable at both 4 and 25 degrees, but they were not stable at 38 degrees. Once we confirmed that the short-term stability of the nanoparticles, the storage stability was correct, we also carried out cytotoxicity assays in order to confirm that the nanoparticles were safe. In order to carry out the cytotoxicity assays, we used the Alamar Blue technique, and we put the nanoparticles in contact with different cell lines. Just as an example, hereby I show you the memantine and memantine nanoparticles results obtained using retinoblastoma cell line. And what we observe is that free memantine was cytotoxic at concentrations higher than 5 micromolar, whereas memantine nanoparticles did not show toxicity at any of the concentrations assessed. This is highly interesting, especially for ocular drug delivery of memantine. Regarding therapeutic efficacy, we studied as well the in vivo therapeutic efficacy against Alzheimer's disease, and in order to do so, we used the double transgenic mice. We had 3 different groups, the control groups, and the two groups of mice treated with the free drug, either ibuprofen or memantine administered orally. And we also had 2 less groups that were the mice treated with either ibuprofen or memantine nanoparticles. Actually, they were treated in the water. The nanoparticles were in the water that they drank, and we left the mice drink during 8 hours, followed by 40 hours of clearance. We carried out several assessments. Some behavioral assessment that we undertook was the Morris-Water maze, where in this test, we put the mice into a swimming pool with a hidden platform, and we trained the mice to find the platform during 6 days. And on the seventh day, we carried out the test. What we measured here was the escape latency, that was the time that the mice took in order to arrive to the platform. And we recorded this parameter. And as you can observe here, actually, we saw that memantine nanoparticles were able to decrease escape latency in a significant manner, both against memantine-free and against the control group. Actually, if we look at the paths that the nanoparticles against and the free drug, the mice treated with the free drug follow, we can observe that actually that the mice treated with nanoparticles, were able to look for the platform in a much direct way than the free drug groups, and as of course better than the untreated groups. Afterwards, we also observed the brain cortex of the mice, and we counted the number of beta amyloid plaques in the brain cortex. And what we observed was that actually there was a significantly less number of beta amyloid plaques in the case of memantine nanoparticles than free memantine and than the untreated group. And the same happened with the inflammation. A similar results were obtained when we assessed the ibuprofen nanoparticles, where the inflammatory state of the brains of the animals was much less with the ibuprofen nanoparticles, and in addition, there were significantly less number of plaques. Afterwards, we also assessed the nanoparticles against glaucomatous neurodegeneration, and in order to do so, we used the OHT model. This model consists on the induction of glaucoma by a hypertonic saline serum injection on the episcleral veins of the rats. In order to do so, we inject this saline serum in one of the eyes of the rats, and we had 3 different groups: a negative control, and then rats treated only with saline serum, and rats treated with memantine-loaded nanoparticles. The treatment using memantine nanoparticles was 2 drops per day during 3 weeks. We recorded the intraocular pressure during these 3 weeks, and what we observed is that memantine nanoparticles were not able to reduce this intraocular pressure. But after sacrificing the mice and extracting the retina, we counted the retinal ganglion cells density, and actually we observed that the nanoparticles, memantine nanoparticles, were able to increase the retinal ganglion cell density against the control, which means that actually memantine nanoparticles were neuroprotectant at the retinal level, although their mechanism was different than the IOP reduction. So as a conclusion, we can say that we have developed and optimized nanostructured carriers. These carriers were stable and were able to obtain a prolonged drug release. In addition, they were non-cytotoxic, and they were effective against Alzheimer's disease and against glaucoma. And just as future perspectives, projects that we are working nowadays, we are working with PLGA nanoparticles targeted with cell-penetrating peptides, as well as targeted with antibodies. We are also working with functionalized lipid nanoparticles, and we're also working with peptide amphiphiles for drug encapsulation. And finally, I would like to thank you all for your attention, and as well as our sponsors and to the University of Barcelona. Thank you very much. And if you have any questions, of course, I'm open to a debate.
Right, thank you very much, Elena, for a nice presentation. So the paper is now open for questions. As I mentioned earlier, if you're interested in posting a questions to Elena, please do so by raising your hand using the application, or by sending me a message through the chat stating your name and your interest in posting a question, and I will give you the floor to make the question yourself. So there is no need for you to write down the question. Okay, so while we wait for questions, let me remind everyone that these webinar series are held every other week on Thursdays at 3:30 p.m. If you're interested in double checking upcoming webinars, you can do so by looking at the CIBERNED website. And also, if you're interested in participating in this series, please do so by sending email message to either myself or Teresa Iglesias, which is the coordinator of training, and we will try to accommodate in a slot for the next few months, a few weeks. Okay, so any questions? Okay, I don't see any question coming through the chat. No raise hands. Please don't be shy if you want to ask a question. Okay, I will make a quick question just to break the ice. Out of curiosity, Elena, what is known about the the actual transfer of this nanoparticle through the blood-brain barrier? Is that different from the system that you can do in vitro? Can you model that in vitro with a relatively reliable way before going to in vivo?
Well, actually more than modulate, what we do is we first design the nanoparticles that have a core that is usually PLGA. And then into the surface, you can attach different kind of ligands. That is what we are working actually now. You can attach either peptides, such as cell-penetrating peptides, and of different kind. And you can also attach antibodies that can provide an active transport through the blood-brain barrier, or different other kind of ligands. And so you can more or less modulate the transport using these strategies. And of course, you can test this in blood-brain barrier, in vitro blood-brain barriers models. And then what we do is we confirm the in vivo, these results with the in vivo results. And in order to confirm the in vivo results, we do two things. Usually what we do is a pharmacokinetic assessment of when we compare either the free drug with the nanoparticles, with the drug encapsulated inside the nanoparticles. And we observe how much drug in each case arrives to different tissues. And also, we also label the nanoparticles. We also label the nanoparticles using some fluorescent markers, and we observe the nanoparticles. We could observe the nanoparticles at the brain level.
Okay, good. Any idea of what is it? I mean, these nanoparticles you administer in the animal in vivo, you administer intravenously, and if that's the case, any idea what is the percentage of nanoparticles to get into the brain?
So in this project that I have presented, we have administered them in the drinking water of the animals. But in the projects that we are undertaking now, we are doing an intraperitoneal administration in order to control better the dose that arrives. And we don't know exactly which percentage of the particles arrive, but that's why what we do is confirm that actually the nanoparticles themselves are able to arrive to the brain. So they are able to cross through the blood-brain barrier. And what we measure is the drug that arrives. But we don't know exactly, or we haven't calculated yet the percentage of drug that arrives to the brain.
Right, so we have a question from coming through the chat. The question is from Gerard Esteruelas. Actually, Gerard, I'm not sure whether you have a microphone and you want to do the question yourself, or because I think you really right. Actually, you wrote down the question. I'm not sure whether you can see it, Elena.
Let me see.
Read it for you.
One moment. Well, it's what I have just said. We confirmed that the nanoparticles are able to penetrate through the brain by labeling this nanoparticle, either the polymer. We label the polymer using a fluorescent marker. In this case, we use rhodamine, but we can use different kind of labeling. And that's afterwards, we injected the nanoparticles and we extracted the brains, and we observe if we see this fluorescence in the brain that it's caused by the nanoparticles.
All right, thank you. Thank you, Gerard. Any other question? Okay, I don't see any raised hands or any message coming through the chat. Let's wait for a few more seconds, a couple of minutes to see if there are some latecomers discussing. No? Okay, this will be the last call for anyone interested in making questions for today. No questions? Okay, so if there are no more questions, then I think it's time to close the session here. Not before thanking Elena for her willingness to share with us her work, for a nice presentation, and thanking everyone for attending the webinar. And I hope to be with you again in a couple of weeks for the next webinar. All right. Have a great afternoon, everyone. Bye-bye. Thank you, Elena.
Thank you. Bye.
Bye.